by mutual agreement, I am just going to introduce Guillaume by saying that he's a group leader at the Academy of Finland. I was going to say he's a new group leader, but he has reminded me that it was 5 years ago. Guillaume, as you are going to see, is an absolutely superlative microscopist, giving a range of beautiful images of cells invading during cancer. And it's also fascinating to me because he has been an early adopter of machine learning as a way of tracking it. And with that, Guillaume, over to you. And thank you very much for kicking off our seminar series.
Yeah. No, thank you very much for the very kind invitation. And I'm very sorry I couldn't join in person this time due to family reason, but I'm hoping that I will be able to come and visit all of you in the future. I'm a big fan of all the work that's been done in UCL. So as Rob introduced, so I'm a group leader in Turku, Finland at Avoakademi University. And today I will be telling you a little bit about some of the work we've been doing, a little bit related to some of the microscopy tool we've been developing and how we've been using them to study cancer cell metastasis. So this is the outline of my talk. Of course, I won't be able to tell you everything we're doing, and I won't try, but I want just to give you a little bit of an overview of some of the topic we are very interested in case you want to be in touch. We try to be very open and collaborative about what we do. And then you will see, so as Rob said, we do quite a lot of microscopy. And because of this, very early on, we've been interested in analyzing data. And for that, we've been also contributing to developing image analysis tools. And I will briefly introduce some of them in case they could be useful for your work. And then for most of my talk, I will be kind of focusing on some really basic cancer cell biology we've been doing to try to understand how pancreatic cancer cell extravasate. All right. So I'm sure I don't really need to introduce this to all of you, but one of the main reasons why cancer cell or why cancer is so deadly is because, especially in solid tumor, cancer cells can acquire the capacity to disseminate. So the cancer cells can leave the primary tumor, where they will first invade through the local stroma, where they will interact with a completely new environment, with new cells, and they will then somehow reach the circulation, whether it's a lymphatic or the bloodstream, survive in the circulation, interact and attach to blood vessel wall at distal sites, cross them, and form a metastasis lesion. So one of the things that to me is really fascinating in this process is how flexible the cells are capable to interact with a very completely different environments and different cells as they undergo the metastatic cascade. And of course, most cells will not be able to make it. Most of them will be dying, perhaps in the circulation. But still, it's fascinating to me to try to understand how cancer cells can deal with this interchanging environment, adapt, and migrate in those different contexts. And this is kind of the question that is driving my lab, where we're trying to really understand how cell interacts with various environments and other cells during the metastatic cascade. And to do that, we are, have a wide range of topics in the labs that we are working on, both on kind of cancer cell biology and endothelial cell biology. We have quite a strong focus on the actin cytoskeleton, and in particular on protrusions, so the type of structures the cells are able to use to sense their environment. And we mostly use imaging methods and image analysis methods to study those data. So early on, I wanted to introduce my lab, who are the people doing all the work I'm gonna present today, with many of the work done by Gauthier Follin, who's been a postdoc in my lab, actually a joint postdoc in my lab, and together with Joanna Iwaska here at the University of Turku. I also want to say that I have currently a postdoc position available in my lab, especially for developing software for analyzing live microscopy data, so that if you're interested in these kind of topics, please don't hesitate to reach out. I will be happy to discuss further with you. Okay, so basically what we've been doing for the past 5 years, and actually me, for much longer during my postdocs and already during my PhD, is to try to understand how cancer cells migrate and interact with their environment. So here you're looking at actually fairly old data now that I acquired during my PhD, where you have an ovarian carcinoma, which has been labeled here in green to visualize the actin cytoskeleton, which is migrating on cell-derived matrices. So those are extracellular matrices generated by fibroblasts that have also been labeled. And I like this movie because it's a really nice kind of illustration of many kind of phenotypes that we are, and it's many other people are also interested in trying to understand, is how a cell in a kind of native 3D environment is able to migrate, how is the cell interacting and sensing the environment. And then also in this movie, as you can probably see, the cell is also eating up a lot of this extracellular matrix here in magenta, and how the cell is remodeling and eating the matrix in order to drive various phenotypes. So we are not interested only in single cell migration. We're also interested in collective behaviors. So this is data that we published a few years back now, together with Joanna Iwaska, where we're looking at breast cancer. So what you're looking at here are breast cancer cells that form the tumor in vivo in the mice. So in a mouse, so we injected the tumors, injected the cells, we let the tumor grow, we took the tumor out and imaged them ex vivo. So the tumor are in their native environment, generated in an animal, but imaged ex vivo. And so here you're not looking at a single cell, but you're looking at hundreds of cells, maybe 1,000, forming the tumor mass. And we were at the time very interested to understand how the cells and their protrusions at the tumor front is important for interaction with the environment. And in this case was to look at interaction and formation of basement membrane. And here now, finally, I just want to show you one last example, which is again a different stage of the metastatic cascade, where you have here, now it's a pancreatic cancer cell in magenta, again, labeled to visualize the actin cytoskeleton. Those have been now injected in zebrafish embryo, and you can see here in green the vasculature of the embryo. And the cancer cell is basically now, it has extravasated, it's left the vasculature and is sitting on top of the vasculature using finger-like protrusions that are perhaps phallopodia, probing the environment outside of the vasculature. So perhaps very reminiscent of a very early event after the extravasation process and the formation of a metastatic lesion. So again, so not going into many details about all of those different type of projects, but just to give you an idea of some of the things that we've been interested in the past few years. And all of that to kind of bring me to the first part of my talk, which is that we use a lot of imaging and live imaging in the work that we do. And if you've been doing live imaging, this is something that you're very familiar with, but it's always about compromises. So you need, as a biologist, for me, the most important is to get from the microscope usable data. And by this, I mean data that you can get biological insight from. But to do that, it means that the sample needs to be healthy, but you also need to get enough resolution, whether it's spatial or temporal, to get the information that you're after while recording your images. And here, really, the most important is the sample health, because if your sample is being damaged, if your cells are dying while you're imaging them, you're really not recording useful information. You're just looking at cell dying on the microscope, which is probably not what you're interested in. So you really need to compromise between those different parameters. But something that perhaps we discuss a lot less about is that you also need to compromise quite a lot when you start analyzing data, because imaging data are typically very difficult and complex to analyze, because the information you want to extract from the images is never going to be the same as from data set to data set. So you always need to compromise between different factors, such as the availability of softwares that may do the kind of analysis you're interested in, and then how accurate they are, as well as the throughput of the amount of data that you need to analyze. And generally, no image analysis solution are 100% accurate. You will always have a degree of errors in the analysis. Even if you do it yourself by hand, you will never be completely perfect. So then you need to compromise between solutions that are available, how fast you want to do the analysis, how much data you can analyze, and how accurate the result will be. And that's something that you need to deal with yourself when you analyze the data, and also be honest with the results that you're getting to make sure that you get the biological insights that you're interested in. Because of dealing with this kind of question, we've been spending quite a lot of time actually developing softwares that are perhaps better adapted to the question that we're trying to answer. It doesn't mean that they're better than other people's software. It just means that they've been designed for our questions. But we've tried to also make them available in case they would be useful for others. So now what I want to do is to show you a little bit about some of the software tools that we've developed over the last 5 years or so. And here, and I say we developed, but it's never been just us developing those software. It's always been in collaboration with others, and I will acknowledge them as I go through the presentation. One of the first things that we had to deal with, and it's perhaps due to our own infrastructure, but when we start to analyze or image large 3D data coming from live imaging from our microscope, we generally have data that is shaky and misaligned. So then we have to spend quite a bit of time to develop tools to help us register and realign our fluorescence live imaging data. So here what you're looking at are pancreatic cancer cell inside of the vasculature of a mouse. So here we've injected the cancer cells in tail vein and dissected the mouse as soon as possible after the injection. We took the lung fresh, we slice it, and we image it straight under the microscope to try to understand how the cancer cells are interacting with the live vasculature of the ex vivo. And when we image those using high-resolution live imaging, there's always shaking because of the microscope, because of the tissue moving, and so on. So one of the tools that we've worked on is called Fast4Direct, which has been very helpful for us to realign 3D data sets, 3D plus time data set, especially when it's multicolor. So if you're facing this kind of problem, you might want to check this out. Perhaps it could be useful for you as well. So Rob already kind of hinted at that. So one of the, I think, biggest revolutions that has happened in the last 5 years or maybe 6 years or so in terms of analyzing image data is the advent of deep learning or AI. And here, when I talk about this, I'm not talking about self-thinking software. I'm not talking about things like the Iron Man suits AI. We can basically do Iron Man's job and do everything. What I'm talking about is software that you can train to do a very specific task and do it very well. So here in the video, I'm showing you one of the video coming from Tesla. So this is as a representation of their onboard computer that is kind of like analyzing the video and live data coming from their self-driving car. So it's figuring out, where the white lines are and so on to kind of dictate how the car should be driving. And so a lot of very similar algorithms can be used now and have been used for quite some time to analyze, biomedical data, including microscopy data. And there, if you're not familiar with those methods, I just put here kind of recap slides on how they work and how different they are compared to more classical algorithms that are non-deep learning based. And typically in non-classical or in non-deep learning based or more classical algorithm, so here is a case for denoising. So imagine you have really noisy images and you want to improve the signal while removing the noise. And what you will typically do is to find what is the best mathematical formula that can remove the noise from your images while preserving the signal without creating artifacts. And this mathematical formula will come from the noise property of your image, which come from the setup that you're using and the microscope and so on. And so they are very good denoising algorithms. So here you have an example which is in wavelets, and you can get really nice denoising. But here one issue that many I would say even experimentalists have is that you really need to have the knowledge in terms of knowing what would be the most appropriate way to denoise those images, right? You need to choose what is the best one to use. When you do and we use deep learning algorithm, the kind of thinking is actually quite different, because there, instead of kind of deciding what is the best way to kind of manipulate your data, what you do instead is that you provide a training data set to a software or algorithm, and you basically tell the software, okay, here are my noisy images, here are my clean images, and then learn how to transform one into the other. And then you feed that to the deep learning models that will then learn from that data set how to apply the best possible manipulation in order to get this denosing. And this is really efficient because and especially as a biologist, for me, it's very attractive because there I can generate, a high quality data set to train. And generally, you can train them to be very, very good, which is typically better than you will get using classical algorithm. And then once you've trained a model, which is kind of more labor intensive, then you can generate prediction from those models and get denosing applied to the same type of images, which can be a very powerful way of processing image data. However, it's not always straightforward to train. So for us, it's really important to be able to train our own model, because the power of deep learning is really to teach the software to do something. So then you really need to be able to train it using our own data sets. So when I started with this maybe 4 years ago or so, I hit a lot of barriers, and so those are the one. I'm not a computer scientist by trade. I was trained as a cell biologist, and it was surprisingly hard to get started with installing all the required software and dependencies and how to connect my graphic card to the right environment with the right things and so on. And then you need to kind of often code, because generally tools that are released come as packages when they are well developed. And so you need to be able to kind of manipulate that. And then you also need to access computing power, because generally you need some at least fairly powerful workstation and graphic cards to be able to work with these tools for the training. So at the time when we got started, it was really feeling like this, where spending days trying to work out why are the software we use, developed by others, why couldn't we use them on our own computers? And it was really a struggle. So I'm going fast here because this is something that we've published already a few years ago, together with Ricardo Enriquez's group. We used to be, I guess, in UCL, so I'm sure many of you know, and also other people in Europe and the world together, kind of almost by accident, started to work on a project called Zero-Cost Deep Learning for Microscopy, where basically we are using a platform called Google Colab, which is, again, something that perhaps you know because it's getting more and more popular, which is a platform that allows you to run Jupyter notebooks in the clouds using resources provided by Google. So the whole idea here, and it's very similar to what others have done, for instance, using Colab folds, if you're doing structural biology, and there are other examples. The whole idea is that we, using those notebooks, provide all the instructions and the code on how to train different type of deep learning model that could be of use for microscopy data analysis. So basically, we provide the notebook. You need to provide the data to train your own model. And within the notebook, you can train model, you can make prediction, you can do quality control, and so on. And so this is something that for us, and especially my lab, has been very useful to get us really started to really use those technologies to apply for our own question, which is cell migration. And so here are some of the things that we could do when we publish the paper. So things have evolved a bit now, but basically any kind of tasks that you can think of for image analysis can be done using deep learning, things like segmentation, using a UNet. So here's an example from electron microscopy. If you're interested in nuclei segmentation, StarDist is a great tool, especially, so we've been using that a lot for automated cell tracking, which has been proven to be very helpful for our own biological question. If you want to kind of identify cellular states, denosing can also be very useful if you do live imaging, because then using denosing tools will allow you to reduce laser power and might mitigate phototoxicity. Something that we've been using quite a lot for live imaging, structural elimination microscopy, is to use things like CARE, which allow us to restore very noisy scene data. And then there's plenty of other applications like super resolution microscopy, either using superpixelation or analysis of those data sets. Something else that we're starting to use more and more, which is artificial labeling, which is to predict one fluorescent marker from another. So here's an example where the actin staining in purple is a real staining, but the green staining is fake and computer generated, which then allow you to maybe multiplex in silico and not having to necessarily acquire all the different signals when you do your experiment. So that was a couple of years ago. It's been very useful for us. We use it all the time. However, as a platform, it has advantages but also a lot of disadvantages that we've been trying to solve. The first one, I mean, that was the main goal of the zero-cost platform, was to try to make it as accessible as possible. What you need is basically internet connection. You need a Google account to use their software, and we can, do some nice quality control to assess how well the models that you train using this platform are performing on your own data. However, then you can start to hit issues when we start talking about reproducibility. So while that's one major challenge that's as a community we need to solve regarding deep learning, is that reproducibility is a little bit challenging, because deep learning model can change over time, and it's not just the models that can change, but the underlying libraries that you need to run them. And we also here in this particular case, because we are using a commercial backend platform, Google Colab, that we have no control over, we cannot fix libraries. We just use the libraries that are currently available in the platform. And because of this, this platform is not overall sustainable, because we cannot control all of it. And it's definitely not private, because it's owned and using commercial tools where we have zero control over any privacy and so on, which may be a major problem depending on the kind of data you're working on. So overall, the platform for us has been wonderful, because it got it started with many of those projects, and it's used a lot as teaching purposes to learn and helping people to learn how to use deep learning. But we've kind of wanted also to solve some of the issues that are listed here. And to do that, again, so working with Ricardo Enriquez's group, and this is work actually done by Ivan and Esti in his lab with a little bit of help from us. We further developed the platform into something called now DL4MicEverywhere, where we are now using containers in order to package each of the notebooks that we've previously developed. So if you're not familiar with those Docker containers, basically they are mini Python ecosystem that are self-contained, which are containing, the correct libraries and dependency to run specific tools, and then you can deploy them fairly easily in different places. So whether it's using your own resources or on a server or whatever you have access to. So here now, using this platform, you don't get any more the free resources that are available provided by Google Colab, but you have now the freedom to very easily install those mini Python containers in order to use those tools on your own resources. And then there you get everything that is associated with it. So then it's fully reproducible and transferable, and it's also extremely private. So then you can do the analysis in whatever private setups that you have to try to address a lot of the issues that we've had with the zero-cost platform. So again, I'm going fast here. I'm just advertising some of those tools. If you're interested, I'm happy to discuss more in detail during the question or later on. So I'll skip this. So the other thing that we're very much interested in my lab, and you probably realize that from the introduction, is we are interested in following cell migrations, right? So we're interested in how cancer cells migrate over time. And the best way to analyze this kind of data using live imaging is to be able to track the cells. So we worked very closely with Jean-Yves Tinovez, who is working at the Pasteur Institute in France, in Paris, with the main developer of TrackMate, some of you might know, which is a Fiji plugin to track. And so what we've done with him is to kind of incorporate a lot of those deep learning tools I've just mentioned to you before, so that they can be directly using TrackMate in Fiji to be able to do those automated tracking. And for us, again, this has been a lifesaver, because now we can really get down to some very accurate and very high quality tracking setup when we are studying cell migration in different conditions. So here you've seen a few different examples of videos, for instance, that you can analyze using TrackMate. And finally, so the last tool I want to introduce that we released a little bit earlier this year, which is kind of following this tracking idea, is that now we are at a stage that we are generating fairly good tracking data using TrackMate and the deep learning tool I was mentioning. And for us, really, or at least for me, the next frontier was, what do you do with all of those tracking data? how can you then combine them, put them together in a way that you can really get a high understanding of the data. So then we started working on a platform called CellTracksColab, which allows to kind of compile and put together CellTrack data in a very kind of automated way, while keeping very easy control over the data structures to be able to incorporate many types of data analysis depending on the things that we're interested in. So if you're interested in kind of cell tracking data analysis, then you could check this out, because perhaps it's something that can be useful for you, or then something that you could build on if you're interested. Okay, so that was perhaps a slightly long first part on some of the tools that we've been working on and developing in the labs to try to solve our problems. But at the same time, I hope that some of those tools will be useful to others and perhaps to some of you. And now what I want to do is basically spend, I should of course acknowledge again all the people that have contributed to this work. So Joanna Pulverleinen in my lab has been driving many of those projects together with Ivan and Esti. And then those are very close collaboration with Ricardo and Jean-Yves Tinvez among many others. But I would say those are the key people that have kind of pushed forwards a lot of those projects that I briefly mentioned. Now what I want to do is to go back to some of the kind of biological questions that we're really interested in, which is trying to understand cancer metastasis and how we've been using the tools that I've introduced to study that. And the things that I want to discuss with you today is the extravasation part of the metastatic cascade. So once cancer cells are in the circulation, how do they get out in order to form metastatic lesion? And to do that, the first thing that the cell needs to do is to survive. And we know that most cells die in the stream. But once they survive, they need to arrest at distal sites. And the arrest can be done through occlusion. So they get stuck in capillaries. And because of that, they are arrested. But we also know that cancer cells can dynamically interact with the endothelium, with endothelial cells using receptors. However, how this is regulated and is still really poorly understood. So this is kind of the first question that when I started my lab, we started to kind of think about is, okay, in the context of pancreatic cancer, how do pancreatic cancer cells interact and attach to the endothelial cell wall, right? So we are not so interested in the occlusion part because this is, a purely size issue. But I'm really interested in the mechanistic into how the cancer cell can actively engage and interact with endothelial cells. And to answer this, we've set up some very simplistic microfluidics setup where we can form an endothelial monolayer. And then basically we perfuse cancer cells on top, right? So it's very simple. It's bright field microscopy, no phototoxicity. And then using this, try to get understanding on how those cells are able to interact. And you probably see where some of that is going, but basically what we've been doing is perfusing different type of pancreatic cancer cell line at different flow speeds. And then we've been using some of the methods that I was showing you to automatically detect and track the pancreatic cancer cells in our bright field video. So the tracking is never perfect, but it's good enough so that we can get a very good understanding of basically the behavior of the pancreatic cancer cells in different contexts. So the way we've been doing it is that we first perfuse at different flow speeds that are similar to what we find in capillaries. Then we slowly decrease the speed and then at the end increase it again to see what are the cells that are actually attached to the endothelium. So using the platforms and method that I've shown you before, basically this is the kind of data we can collect. So here you have the example from 3 different pancreatic cancer cell lines, where you have the PANC 10.5 in green, and you can see that those cells are really bad at attaching. So it doesn't matter the flow speed that we are using. So H is for the highest flow speed, medium, low, and the wash at the end, which is the high again. You can see that those cells are very bad at attaching. We are using also SPC1, for instance. So those are not very good at attaching at the beginning when the flow speed is too high, but then if you decrease a bit, the cells can attach. And then we have here a third example, which is the MIAPACA2, and then those are able to attach regardless of the flow speed. So basically, and this is something that is intuitive and was also known in the literature, is that, of course, different cell types will have different capabilities to engage with the endothelial cells, but also that flow speed is important to modulate the attachment. We're also interested to compare the ability of cancer cells to attach with endothelial cells to that of specialized cells. So if you think about immune cells, so immune cells' job when they get activated is to interact with endothelial cells. So they are really specialized for that. So then we repeated similar experiments in immune cells with and without inflammatory cytokine in order to see how does they compare to the cancer cells. I'm cutting a long story short here, but basically this is the kind of data again we get. So we repeated cancer cell with and without IL-1 beta to mimic inflammation, to neutrophils and monocytes. And basically what we found is that neutrophils, when they get activated with IL-1 beta, they are capable of attaching very well to endothelial cells, which is not surprising. But what was slightly more surprising to us is that basically MIAPAC-2 cells, the cancer cell, which are hijacking those pathways, are basically showing very similar adhesion profile to those of immune cells, showing that those cells are definitely very, very good at attaching with the endothelium. Something that was a little bit boring for us is that here in this particular case, the inflammatory cytokine didn't really change very much how the cancer cell were interacting with the endothelial cells compared to no treatment at all. But the main difference between cancer cells and immune cells in our hand is that the cancer cells are not able to cross the endothelial monolayer within the 10 minutes of imaging that we have, whereas the immune cells, as soon as they attach, they're very good at crossing the barrier. So even though they might have similar attachment profile, the extravasation part, or here transmigration part, is different. So here you've seen an example of both monocytes and neutrophil. They can attach and cross straight away. So the main difference that we've seen in our hand comparing specialized cells and cancer cells, so even so it's kind of comparing apple and oranges, the main difference is not in their attachment profile, but in their ability to cross the endothelial layer within those 10 minutes of activation. Okay, so what I've shown you so far is that we've worked on developing a quantitative and imaging analysis strategy to quantify how circulating cells attach to endothelial cells. I've shown you that pancreatic cancer cells display distinct RS behavior that are sensitive to flow speed. They are as good as immune cells, broadly speaking, to interact with endothelial cells. At least in our hand, in this case, the pancreatic cancer cells are not very sensitive to IL-1 beta stimulation. So it changed a little bit the attachment profile, but not very much at all. But then, so this is nice, but now we're doing microscopy. So can we learn anything else? Can we get any special information from our live imaging data? So the first thing that we did was we thought, okay, we have spatial information. Can we get subcellular resolution in where the cancer cells are arresting and interacting with the endothelial cells? And so this is where we start to use again some of the deep learning image analysis strategy I was mentioning, especially the artificial labeling. So here you have the bright field data, and from there we can identify the circulating cells and we can track them. But we can also, from the bright field data, predict what cell-cell junction and nuclei staining will be like using, in this case, pix-to-pix, so artificial labeling tools. And then from there, segment the endothelial cell as well as their nuclei with a fairly high degree of accuracy. So when we do that and we can combine it with our tracking data, then we can start to get information about where are the cancer cells or the immune cell landing on the layers. Are they close to a junction or to a nucleus? And when they arrest, is it close to a junction, a nucleus, and so on? So the pipeline is working really well. Unfortunately, the results are a little bit boring in the sense that regardless of the condition we've looked at, whether it's cancer cells or immune cells, the cells are always arresting very close to a junction, if not on top of a junction, and further away to a nucleus. So from all our data, whether it's cancer cell, immune cells, different treatments, we always see the same result, is that the cancer cells arrest on top of a junction and further away to a nucleus. So We can also see that in the fixed data, and it's true across most cancer cell lines, if not all the cancer cell lines we've tested. Cells, when they arrest, they are on top of the junction and not on top of a nuclei in 95% of the time. So what does it mean? So that's something that we thought quite a lot about. And one of the things that we've looked at is whether or not there are some topological reasons why the cells will be arresting there. So we did some atomic force microscopy using scanning mode, where we look at the topology of the monolayer and to see whether the nuclei would be basically a hill and the junction would be more of a valley. And actually, there's a very little differences between in height differences between the nuclei and the junction. The difference is about 2 micrometer. And the distance is between two is about 40 micrometer. So the slope is actually fairly small. And if you think about a cellular scale, 2 micrometers is very, very little. We also look at the stiffness and the mechanical measurements of those endothelial monolayers. And again, so here what we found is that perhaps as expected, the endothelial cell-cell junctions are a little stiffer compared to the rest of the endothelial monolayer, so the nuclei and cytoplasm, by about 20%. So there is definitely a difference, but we don't think that this is enough to explain why the cells will be arresting on top of an endothelial cell-cell junction. But perhaps even more important is, what does it mean for the cancer cell? So what we see is that when the cancer cells are sitting on top of a junction, and here you're looking at structural elimination microscopy, 3D in fixed data, when the cell is sitting on top of an endothelial monolayer, so here the cancer cell is in purple, stained for the actin cytoskeleton, and the endothelial monolayer is in white, and underneath in green, you see fibronectin, so extracellular matrix molecule fibronectin. What you see is that the cancer cell is not in contact with fibronectin on top of the layers, but because it's sitting on top of a junction, it provides access to the underlying substrate, and we can see phallopodia protrusion in this case crossing through the layers, which may contribute to the extravasation step. So we think that the fact they are on top of a junction may facilitate downstream transmigration of the cells. The other thing that we've noticed is that when we look spatially where the cell arrests on the layer, it's not uniform. So here you're looking in those images at each of the dots represents the location where a cell will be arresting on top of a layer. So if we start looking at them as points, you can see that it's not homogeneous throughout the layer. You can see clusters and patterns in where the cell is arresting. So it's definitely not random. And we can quantify that using replay score to look at clustering effects. And some of the cancer cells are very good at clustering, especially SPC1, at clustering on top of an endothelial monolayer. So there's definitely factors influencing the arrest of the cells in those areas. So we looked into that, and again, to cut a long story short, we looked at a lot of different receptors that may be expressed by the endothelial cells in a kind of a patchy pattern. So looking at, for instance, again, fibronectin, because there was evidence from literature that maybe cancer cells will arrest on fibronectin on top of the endothelial cells. We look at ICANS, which are important, cell-cell adhesion receptor, as well as CD44, which is also an important ECM regulator. And what we found is that actually pancreatic cancer cell, or 95% of them, arrest on CD44 positive cells, whereas it's only about 20% for fibronectin and ICAM. So there seems to be a strong correlation between where the cell arrests and the expression of CD44 on top of the endothelial monolayer, at least much higher than will happen just by chance. But for us, this, of course, correlation is not causation. So we wanted to kind of get a little bit more of a grip on this. So first, what we did is to try to simulate, right? So we wrote a very simple simulator, and I'm happy to discuss more about that if you're interested, where we basically in silico perfuse cells on top of a layer of cells. And then basically here, the arrest is basically only probabilistic based. So then you can assign a probability based on the adhesion strength of your cells to attach or not attach. And then you can change that probability based on, for instance, CD44 expression level in the background to see whether or not these kind of things would be sufficient to drive clustering based on real microscopy data and to see whether or not it mimics what we see in our live experiment. And basically, the answer is no. So at least in those simulations, we couldn't repeat what we see in our live imaging data by just adding, a higher probability of attachment of where the CD44 is on the endothelial monolayer. So in silico, the only way that we can start to really see clustering is when we take flow into account. So as soon as we start putting in our system that the attached cells will be locally deflecting the local flow around already attached cells, then you can start to see clustering. So basically what it means is that at least in our simulation, when a new cell attach, we see changes or large changes in the local flow fields, which then will be impacting the subsequent attachment of cluster cells. And this trigger actually a very high degree of clustering in silico. So we think that altogether, what matters is not only the presence of a specific receptor on the endothelial cells, but also local flow fields will be also contributing to the attachment of the cancer cell on top of the endothelial cells, at least in this kind of experiments. So these are the results now of the experiment. So when we take into consideration a dynamic flow field, by this we mean that we can allow for changes in the flow field when cells attach. We can start to see very high degree of clustering, and especially at high flow speed, which is exactly what we see in our live imaging data as well. Okay, so from the spatial analysis, that what we found is that the cancer cell arrests in proximity of endothelial cell junction, perhaps because of stiffness. But I think most importantly is that it provides them access to the underlying extracellular matrix, and we can discuss that if you're interested. We've observed that the PDAC lands on hotspot on top of endothelial cells, and they are CD44 positive. But at least in silico, it doesn't seem enough to have CD44, or at least our CD44 staining are not sufficient to explain on their own these clustering effects, unless we start taking into consideration possible changes in flow dynamics around arrested cancer cells. Okay, so you're going to tell me, okay, that's all nice, but what about any kind of molecular mechanisms? So basically, this is something we've been trying to tackle for quite some time, and one of the ways that we've been doing this is by training cancer cells to be better and better at attaching to endothelial cells. So again, cutting years of work into one slide. But basically what we've been doing is perfusing cancer cell on top of endothelial cells and then purifying them again, and then doing it again and again until we start producing, population of cancer cells that are better and better at engaging with endothelial cells. Then we did single cell RNA sequencing to see if there are changes in their gene expression. We've identified about 40 to 50 genes that seems to be upregulated in cells that are very good at attaching to endothelial cells. And then to narrow it down, we did a mini siRNA screen. So to develop this kind of things, we had to fix quite a few challenges. So the best option we found was to use cellular barcoding, where we can mix population and color them in different colors to see how different population of cell can engage with endothelial cells. Again, if you're interested in that, I'm happy to discuss it. And interestingly, one of the top upregulated genes is CD44. So kind of going back again to the CD44 that we've already observed to be somehow related to the attachment on the endothelial cell. So I'm not going to show you a lot of the results from the screen itself. Instead, I want to show you some of the work we've done on CD44. And basically what we did is that we targeted CD44 both in the cancer cell and the endothelial cells using a siRNA or blocking function antibody. What we found, and you will see in the video here, is that both from MIAPACA and SPC1, as soon as you target CD44 in the cancer cells, you completely stop the ability to attach to the endothelial cells. So you stop the ability to cluster with each other, and you stop the ability to attach and engage. So CD44 in the cancer cell seems to be very important for the initial attachment in our hands. And so here are the quantification for this. So the data are very black and white. If you silence CD44 in the cancer cell, you completely stop or almost completely stop the ability of the cell to attach in both MIAPACA and SPC1. And we can do that using a siRNA or with blocking function antibody. And I'm not showing those data here for the interest of time. However, if you do that in the endothelial cells, actually nothing happens. So targeting CD44 in the endothelial cells is, first of all, challenging, but from the degree of what we can do, it doesn't seem to affect the ability of the cancer cell to attach. So the other thing we've looked at is CD44 binds to several things, one of which is hyaluronic acids, or HA. So then we've digested hyaluronic acids in our endothelial cell and in the cancer cell to look at how digestion of hyaluronic acid will impact attachment. And again, so if we remove HA, either on the cancer cell or on the endothelial cells this time, we completely stop the ability of the cells to attach to endothelial cells. So it's, again, very clean experiments where if you don't treat, you have nice attachment of those cells to endothelial cells, but as soon as we remove HA, either on the cancer cell or on the endothelial cells, we completely block the ability of the cell to attach. And so we're still very much interested to study how this is regulated and exactly what happens and what kind of adhesions are formed between the cancer cell and the endothelial cells, but that's still very much work in progress. So this is now my second take home. So I've shown you that PDAC can interact with endothelial cells quite well and as efficiently as circulating immune cells. They are arrest and land in the proximity of cell-cell junction, which may provide access to the ECM and contribute to further down extravasation. I've shown you that PDACs arrest on hotspot on endothelial cells, and that CD44 and HA modulate PDAC attachment on the endothelial cells. But then you might ask me, okay, so now you have attachment, but what actually happened in terms of extravasation? So how does the pancreatic cancer cell, transmigrate? So this is obviously something we are very interested in as well, and I will just give you a small teaser of some of the experiments we've been doing for the last 3 years on the second step of the extravasation process. And then for that, we've been, again, going back to live imaging at high resolution to look at how the cells are interacting with endothelial cells. So here you can see PANC10 cells that are not great at attaching, but they are also not great at extravasating. So here you have the endothelial cells in magenta, and the cancer cell, again, labeled for the actin cytoskeleton in green. And you can see that the PANC10, it's sitting on top of the junctions, but it's not really able to do very much. So it doesn't matter if the junction actually opens up a little bit and you get a gap, the cells are not able to cross. Now I will show you what happened with MIA-PACA, which are MIA-PACA2, which are very good at attaching and crossing. And what you see actually is that the cell is again sitting on top of a junction, and it's extending those protrusions that I've already shown you using structural illumination microscopy. And those protrusions can cross the endothelial barrier to contact the underlying ECM. And we think that this step is very important to trigger the transmigration step here in this case. And then now you have the cancer cell that has crossed the endothelial monolayer. So now we're very much interested to understand how some of the receptors that I've described, for instance, CD44, is linked to some of those steps, but also whether some of the other receptors involved in cell ECM adhesion, and what happens if we block them, and what happens if we target some of the cytoskeletal regulator involved in phallopodia regulation, for instance, and how can we block this transmigration step. But this will be a story for another time. Okay, so now I would like to acknowledge many of the people that have contributed to the second part of my talk. So most of the work that I've shown you have been driven by a very talented postdoc, Gauthier Follin, with a joint postdoc also in Johanna Ivasca's groups, together with Suzanne Guimaya, who's been doing a lot of those cell experiments that I was showing you. So Johanna has been working very closely with me together on the data analysis for this project. So Monica has worked on the screen part that I briefly mentioned. And then this was fantastic collaboration with Johanna, but also with Marco Salmi, who's been teaching us how to purify and how to work with primary immune cells for this project. So a lot of the thanks goes to them. I also want to acknowledge funding, because without any funding, I will not be able to do any work. So we are lucky to be funded by many foundations that are supporting our work, and for that I am very thankful. So on that, so this is everything that I wanted to show you today. Hopefully I have not overrun too much, and there's still time for question and discussion if you have any. And as I want to further advertise, so we still have a postdoc position available in my lab. So if any of you are interested, please be in touch. Thank you so much for listening, and I'm happy to take any question.
Thank you very much indeed, Guillaume. Could everyone please unmute and clap, or use clap emoticons, or find a way of showing Guillaume your appreciation, please? Let's see what we can see. There's one. If you want to ask a question, you can go through the chat, or you can put up your hand. And I'd very much like to say everyone, I saw a hand up, but it's gone. Maybe that was just a clap. I'd be delighted to have any questions. Anna is a group leader. I wonder if any of you students and postdocs who are there can ask a question first before we get to Anna. Oh, go on, be brave. Let me see, I'm looking at different, hang on, this is the bottom.
There's a question in the chat if you want to.
Yes, let's go for that. Veronica, do you want me to ask a question, or do you want to unmute and ask it yourself?
I think that was an amazing talk. We really enjoyed it. I was
Thanks.
wondering why you chose PDAC in particular as your cancer cell model.
Okay, so I guess there's this maybe 2 or 3 levels to answer here. So the first one is obviously pancreatic cancer is a devastating disease, and there's a lot of things to learn still, I think, on how the disease progresses. And there's a lot of opportunity here, I think, to develop new therapeutics, right? And really help patients. So that's the first level, which is perhaps the most political one to answer. The other one is that there's very few people in Finland who works on pancreatic cancer, which means that there's a very nice niche to work in to get funding. So those are the 2 main reasons that I've decided to kind of move toward that, because we have a lot of people, for instance, working in breast cancer, in breast cancer cell biology. So then the field is much more crowded.
And it's a lot of deaths, isn't it, Guillaume?
Yes, yes. So in Finland now, the number of people dying of breast cancer and pancreatic cancer is roughly the same. But it's, pancreatic cancer is on the up, whereas breast cancer is getting better and better. So in the next couple of years, there will be more people dying of pancreatic cancers as there will be people dying of breast cancer in Finland.
Thank you so much.
Yes. Anna, what's your question? Anyone else, please put your hand up or put something in the chat. Anna.
Yeah. Hi, thanks for your nice talk and very interesting. I was wondering about the cells landing on junctions. I mean, I don't know that much about cancer cell extravasation, but in other kinds of cell invasion, they often show that it's at tricellular junctions where it's a bit easier to invade. Is that true for you too, for your system?
So we've looked at that as well, and it wasn't that clear. I mean, there's definitely cell landing on tricellular junction, and actually in one of the videos I've shown you, it's really on top of the tricellular junction. But we have example of both, and we've not carefully quantified to see if there will be a preference toward tricellular junction versus normal junction. Other people have shown that tricellular junction are maybe preferred, but I would not be able to tell whether that's the case for us or not.
So I mean, also I was wondering, when you say that you see them landing on the junctions but not in the middle of the cell, I don't know how your time resolution is, but could it be that they're transiently landing somewhere else and then they move further towards the junction?
So what we see is that they are always landing closer to a junction than a nucleus. So that's more accurate way to describe it. And our time resolution is actually pretty good. So we image every 20 milliseconds.
Oh, wow.
So then we have fairly good resolution of where they land. So it's not to say that they wouldn't land ever on a nuclei, but overall they land closer to a junction than to a nucleus. And I think there, to me, part of the answer is also crowding. So there's many more junctions on the field of view than there are nucleus or nuclei in this case. So it's more likely, statistically more likely to be close to a junction than a nucleus.
Okay, thanks.
We have no more hands up, so I'm going to ask the first of my many questions. But I'd be very happy if people other than me thought of things to ask. Guillaume, I'm hoping that we will get quite a lot of people at UCL using TrackMate 7 and CellTracksColab. But can you tell us what the key features in the microscopy are? What makes it work better and what might stop it from working?
Yeah, so I think for us, the main reason why we started to work on this is, I mean, you know, as a cell biologist, there's a lot of features that I was missing from existing software.
We know.
So I think the main thing that I wanted to work in on TrackMate was better segmentation to get more accurate tracking. So that was the first thing. The other thing was batch processing. So for me, it was always a nightmare when you have a tracker that works well, something with a graphical interface that you can start processing large amount of data. So this is something that we were actually quite hard in TrackMate 7 to incorporate. So those are actually the main reason why we've been working on this.
But what makes it go wrong? What causes artifacts? Or what should we do to not get artifacts?
So typically in tracking, the most likely reason of causing artifact is, especially if you use a tracker like TrackMate, where you have first segmentation and then linking between frame, is poor segmentation. Typically, this is the biggest issue, is that if you don't get a very good segmentation, or if you have missing segmentation, then it would be hard for the tracking tool to do proper tracking. So this is why also for us it was so important to have the possibility to use deep learning for the segmentation parts of the tracking to get the most accurate possible tracking.
Right. And the deep learning is uniformly good, or there are particular things that confuse the deep learning? Is it, if you get a pile of cells, does that ruin everything, or? Well,
so with deep learning, I mean, for us, the important is to be able to use our own models that we train with our own images, so that we can get, you know, the most accurate possible segmentation.
Okay. So the secret is self-training with your cells in your microscope.
Exactly. So I mean, you can have some really good deep learning models that might work straight away on your images, depending on what they are. So for instance, we've had very good success with CellPose, especially the latest model, Cyto3 is very good in many other cases, but then typically when it starts to not work anymore, then you need to fine tune it on your own images to be able to get the best possible segmentation.
Thank you. Iona, then Barbara. Iona first.
Hi, this was a great talk. And actually, I want to follow up on a question that Anna had. So she asked, you know, whether those cells that attach on near particular junctions. Have you looked at the cells that attach, like, to see whether they have any common morphological features themselves? I mean, are they of a certain sphericity or size, or do they have?
So I mean, we you know, when the cells that attach, we don't see any major differences, you know, per se in terms of morphology. So the cells when they attach are very round, and they're all, you know, very, very similar. From the RNA sequencing, you know, we can tell that there are small differences, but only when you start training them. So like when they get better and better at attaching, then you can see a shift in the population. But then when we did single cell RNA sequencing between cells that have attached versus either the parental or cells that are not attached, we cannot see any difference whatsoever, even in gene expression. So my guess is that, you know, the attachment part is such a random process, because very few cells are capable of attaching, and among the ones that are attaching, they're only the ones that are within the plane of the attachment. So at least there, we couldn't see any, you know, specificity. But here we're working with cell line, right? So then if you think about now, you know, patient, for instance, where you will have a very high heterogeneity from the primary tumor, then I'm sure there that you will have, you know, from that heterogeneity, you will have cells that will get expression of specific genes that will drive the attachment and make them better than others. If that answers your question.
I mean, no, it's good. I mean, those changes could be at the post-transcriptional level that you don't see with the RNA.
Sure. So that we don't know.
That's great.
Yes, that we don't know.
Thank you. Perhaps the last question that we have time for is Joanna.
Hi. So I have two questions. One is more methods and the other is more on biology. So I don't know if I missed it. What would you say is on average your sample size for training for the cell migration and tracking behavior?
Good question.
Good question. So this is a question that I get all the time, and it's also one that is very hard to answer because
it depends on the
so
sample.
much on the data.
Yeah.
So with deep learning, as always, it depends what you want to do. So we, at least in my lab, we are a big fan of over specialized models that answer a very specific question. So for some of the data that I've shown you, I think we've trained 10 or 12 different segmentation modules because each of them, answer a very specific question with a very specific type of data. And then there, if you want to do that, then it depends on how variable your data is. So then if your data is fairly uniform and you have a lot of examples in each of the images, you can start to get quite good, model from maybe 10 or 15 images. So you don't necessarily need a lot of images if you want something that's very, very specific. Now, if you have data that you want to analyze with the same model that is very variable, then you need to start to provide during training representative images that represent the variability of your images. And then the number of images depends on how variable your data is. So then, we've had models that we train with maybe 50 or 100 images. And then the next level is that if you want to train a model that will be, as a deep learning model, useful for the communities that will work across microscopy modality, across image type, then you need to start training with much, much bigger data sets. So it depends a lot on what your objective is.
Yeah, I see the point. And then my second question about the biology is, so you mentioned as well that cells, when they go into circulation, they don't survive as well, and
Yep.
they need to adhere in an area. And then, so would you consider that this adhesion is actually crucial for their survival, or are they already kind of winner cells that they can survive, and this is therefore why they adhere in the junction area?
Well, so in vitro, in our in vitro experiments, so we don't really have any cell death within the time that we do those experiments, right? So the cell survival there is very good. Now in vivo, in a kind of a tumor context, I would say, I think from what people have reported is about 95% of the circulating cells will die, and only very, very few will make it, through the metastatic cascade. It might be even, less than 1% that survive. And there, I think it's, in my opinion, is that it's probably both, right? So the ones that make it are the ones that will have somehow managed to survive, either because they've have mutations that protect them against anoikis, or then they are forming clusters, and then they have pro-survival signals coming from cell-cell interaction, and they can also attach. So then they can cross. So I would think that both are important.
But then, do you ever see whole clusters? I mean, like from what I saw in the movies, it seems more like there is going to be initially one cell that is going to adhere, and then more of them will come over. So do you ever see a whole cluster of, I don't know, 3 or 4 cells actually adhering there?
Yeah, so we do see small clusters, rarely attaching, where we have maybe 2 or 3 cells that will be attaching together, because this is much, much rarer. So we see much more the other way, where we'll have a single cell that attach and then clustering that happen afterwards. Yeah.
Okay, very, very interesting. Thanks for the talk and the answers.
Yeah, you're welcome.
I think we've gone over time, Guillaume, so I think we should allow everyone to go now. Thank you very much indeed. That was excellent.
Yeah, thank you very much. Right, nice meeting you, everyone.